Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Dr. Beth Shapiro, who is the Chief Scientific Officer at Colossal Biosciences. You may have heard of Colossal from their recent de-extinction of the dire wolf and also plans to bring the woolly mammoth back. Beth has written multiple best-selling books on the topic of how we can think about all of this work in the context of how humanity has shaped evolution over time, what the tools and technology that we have in front of us today. And I'm really excited for this conversation because I think we're going to learn a lot about an area that we don't often cover on this podcast, which is all those non-human animals that Beth and her team spend so much time thinking about. So Beth, thank you so much for taking the time to be with us today.
I'm looking forward to the conversation.
I want to actually start with how you got interested in this in the first place. Did you have an origin story or a way that really got you excited about combining deep technology and genetics with with wildlife and our understanding of zoology more broadly?
I think my origin story is always disappointing to the people who ask me that question. I started off in broadcast journalism. I wanted to be a storyteller and talk to people about world events, what's happening out there, and how we can think about how we position ourselves in the context of everything else going on in the world today. And I took a class early in my undergraduate career. It was a field program. I was at the University of Georgia in the honors program, and it was a field class in geology and anthropology. And it was 9 weeks camping outside. We started on the East Coast, drove across the country, drove up the West Coast, drove back across the country to Georgia, slept in national parks, and learned about the history of North America by experiencing it, by sitting there. We learned about the formation of the Tetons by pulling over to the side of the highway, pulling a bunch of folding chairs off the top of our vans and sitting down with the actual mountains behind us and pointing to the different parts of the mountain as we were learning about it. It was pretty amazing. And I thought to myself at that point that maybe this is the type of story that I wanted to tell. I wanted to be a science journalist rather than just a regular journalist. And if I wanted to do that, perhaps I should get a degree in science. So I started studying science and I never really turned back. I didn't do any genetics, really. I took a class in genetics, but I never wielded the pipette until I got into grad school. And I went to Oxford University and met early in my time there a man called Alan Cooper, who was just setting up an ancient DNA research lab. It was one of just a few that were in the world at the time. And he was using DNA from fossils, not really fossils, because if they were actually fossilized, they'd be rock and there would be no DNA remaining, but subfossils or just really well-preserved bones to try to understand and reconstruct past ecosystems. And I thought, this combines my love of geology and paleontology and history and storytelling with what I really want to do is evolutionary genetics. And that's how I got into it. So it's a convoluted story about changing your mind and taking risks, I guess, but never really what people who ask that question are looking for.
I think it's great. And I think it is an amazing through line because you've taken that, I think, original interest in telling stories. And the work that you're doing is about telling some of the most amazing stories through the lens of genetics, some of the most amazing evolutionary stories, but also you do a great job communicating it to the public through some of your books that we'll talk about. But I want to actually talk about that first project that you worked on at Oxford. You were working, my understanding is on trying to understand the evolutionary history of the dodo, and maybe you would describe it as is there a way for us to bring back the dodo? I'll let you talk a little bit about that project, but that must have been really interesting at the time, and it's hard not to get excited about a project like that. So maybe you can tell us a little bit about it and how that influenced you going forward.
You know what's funny about that story is I didn't start working on the dodo. My first project at Oxford was about American bison. And this is because, so at the time that I started, it was 1999, and there were, as I said, just a couple of labs doing ancient DNA around the world, but they divvied up all the coolest animals, and like mammoths went to Svante Pääbo and Hendrik Poinar and that research group, and Bob Wayne was doing horses, and giant bears and lions and the things that people are interested in went to other people. And, obviously Svante Pääbo's group took Neanderthals, and there wasn't really anything cool left. And somebody said to me, you can work on bison and you can work on reindeer, caribou. And I was like, that sucks. Like, I don't want to work on those things. But actually it was really lucky because bison still are alive, there was a lot more power in the types of genetic analyses that we could do than there were with species that were still extinct, because we had really closely related, because it's the same species, reference genomes that we could map to. We could see what happened. So bison, it's actually a really interesting story about bison that I wanted to start, because we thought with ancient DNA, we were going to be able to reconstruct the timing of these declines, the bottlenecks that go toward extinction. And we know that bison nearly went extinct at the turn of the 20th century, when there was this massive campaign to drive them to extinction, really as a means of driving to extinction the Native American communities, which unfortunately didn't happen. A lot of the conservation legislation we have today was intended to protect bison and passenger pigeons, but that wasn't as good of an ending story, really. So we knew that there was this near extinction around the turn of the 20th century, and I was hopeful that I could use ancient DNA from bones that were older than that to show that there was a ton of diversity prior to this near extinction, when there were millions of bison across the American plains, and then see that decline toward extinction and eventually bottleneck and then recovery. Because we know there's no diversity today. So I went back, collected a whole bunch of bones that dated to the last 10,000 years, extracted DNA, and found that there was very little, if maybe no evidence of that near extinction in the 20th century, because in fact, bison had nearly gone extinct at the same time as we lost things like mammoths and the giant bears in North America and things like that. So bison narrowly avoided extinction, not once, but twice in their recent evolutionary history. And if I got DNA from bones that were older than that, I did see this huge amount of genetic diversity. So there was a huge population of bison during the ice ages. And then there was a crash starting somewhere around 20,000 years ago, associated with the peak of the last ice age, a really big crash, then a recovery. And then because there was already very little genetic diversity, we didn't really see that second crash at the turn of the 20th century, as there was already very little there. So that was my first Oxford project, and it was really motivated by trying to develop the tools that we need to be able to do population genetics on samples that are on DNA sequences that are sampled over time, if that makes sense.
Yes. Yes. Where did the dodo come into this?
So our ancient DNA lab was in the Oxford University Museum of Natural History. And every day when I walked through the museum, I don't know if you've been there, but if you haven't been there, you should check it out.
I have actually, yeah.
And in the back of the museum where they have all the human stuff, there are these drawers. You just open the drawers and you look and see these crazy, it's an insane place. Like, strong recommend for the museum there, both of them, they're pretty cool. Anyway, as I would walk back to our ancient DNA lab, I would pass the dodo specimen. And at the time, nobody even knew exactly what type of bird the dodo was. There were lots of different hypotheses about it that were more or less based on morphology and the way people imagined the dodo looked, but no one really knew. And I really wanted to get DNA from that sample. And so I would ask, could I sample this dodo? And they were like, are you kidding? No, absolutely not. This is our prime sample, and you're a first-year graduate student.
Yeah, come back next year.
Yeah, right. So I did work on some of the other samples that were in the collection, toe pads, from other extinct birds. And they finally gave me permission to sample DNA from that dodo. And I actually didn't get anything from it. They let me take the skull, and apparently that skull was thrown into a fire at some point, and then they rescued it. A well-meaning curator rescued it. So it was kind of burned on the outside. They let me take a big long tweezer and sort of dig through the inside of the skull to see if I could pull out any gunk. And no surprise to anyone, I could not recover any ancient DNA from partially charred
skull. Yeah.
skull tongue gunk. I don't know what it was. They did eventually let me take a tiny piece of the leg bone. And I was able to get some mitochondrial DNA. This was published in the early 2000s. We were mostly focused on mitochondrial DNA. The whole field of ancient DNA was, and that's just because there are many more copies of the mitochondrial genome in every cell than there are copies of the nuclear genome, which means that you just have a higher likelihood of getting mitochondrial DNA if you don't get very much DNA preserved in a sample. Now, with the transition from Sanger sequencing to next generation sequencing, we regularly get nuclear DNA from ancient samples, but the fragments that are preserved are really short, like on average 30, 50 base pairs short. So if you imagine having to use Sanger sequencing, you got 2 primers, that's already 40 base pairs, and that's already longer than most of the fragments that are preserved in your sample. So we really needed next generation sequencing, or just shotgun sequencing, to be able to break through into the world of paleogenomics. I digress. I've gone in a lot of directions there. Sorry about that. Bring me back to where you want me.
Yeah. So if we think about all of this focus, it sounds like was really around understanding, right, the evolutionary history of these animals. If we get to where we are today, the technology has moved on, and maybe just our ambitions have moved on as well. We can think about not just understanding, but also resurrecting versions of these extinct or near extinct animals. I'd love for you to just talk a little bit about that, like how you think about the structure and framework of science in this realm. And then we can start to dive into some of the examples, because I think it's important to set the scene a little bit first.
Yeah, so it's interesting. I think the idea of de-extinction, of resurrecting extinct species, has existed since the beginning of the field of ancient DNA. In fact, when I was researching for my second book, Life as We Made It, I used the New York Times Wayback Machine, which is amazing. I love it.
Yes.
It's fantastic. And I was able to read the first article that was ever written about the first ever ancient DNA sequence that was recovered. And it was a paper that was published in Science in 1984 by a team from Alan Wilson's group. Alan Wilson had a lab at UC Berkeley that was called the Extinct Species Study Group. And he was working on museum-preserved specimens of an animal called a quagga, which is an extinct subspecies of zebra. And they were able to recover, using molecular cloning and old school sequencing, right, a tiny little fragment of mitochondrial DNA from this quagga and show that it was closely related to a zebra, which is not that shocking, given that if you look at a picture of them, the skin was about 100 years old, it looks like a species of zebra. So that was not surprising. What was surprising is that DNA was preserved at all after death. And this really set off in motion this idea of, wow, what can we do if DNA survives after death? But the reporter who covered this story became the very first person ever to ask the very first ancient DNA researcher about the very first ancient DNA paper what this meant about whether we were going to bring mammoths back to life. So I think the idea, it's interesting, right? It's a fascinating idea. Can we actually do this? And in fact, when Michael Crichton wrote Jurassic Park, which came after this, a lot of people think that
9 or 10 years later, right? He had a lot of time.
The writing of the book was around the same time, but he in the very first book, he said he thanks Alan Wilson and the Extinct Species Study Group for giving him the idea for Jurassic Park. So a lot of people think that my field was caused by Jurassic Park, but in fact, Jurassic was brought about by ancient DNA.
Yeah. I'm not going to ask a question about Jurassic Park because it's probably just the most, you probably get it 100 times a day, but
I don't mind it though. I think it actually is a really nice grounding way to talk about the technology and what we're really doing. But just to finish that loop there, and as I was working in the field with bison and with other species and learning how to do this, we would publish papers and always be excited to talk to people about it. But the first question we always got was what it meant about bringing extinct species back to life. And in fact, that was the motivation for writing my first book, which is called How to Clone a Mammoth, which really is intended to be a kind of instruction manual for all the technologies that we would need to develop to have de-extinction be a realistic thing. So that's how we got there.
Yes. I want to spend most of the episode understanding how to clone a mammoth, but if we just start with why first, the dire wolf, the mammoth, maybe you can talk about some of the work you're doing at Colossal and what is the counter argument is we may be messing with fragile ecology if we were to ever bring these animals back. Maybe you can make your argument of what is in it for us as society to spend the time and resources to do this.
So I have spent a lot of time in the Arctic collecting bones from things that used to be alive and thinking about how we can use this technology to learn about what makes some organisms more robust, more resilient, excuse me, and what makes some ecosystems more robust and more resilient. And my goal has always been to use the past as a kind of completed evolutionary experiment to make more informed decisions about how we can protect and preserve ecosystems and populations and species today and into the future. And as I've been working in the Arctic, what I've seen firsthand is the acceleration of the degradation of these ecosystems. Obviously, the Arctic is impacted much more quickly than other ecosystems because the pace of ecological change there is faster. And I've seen the work that Sergei Zimov and his son Nikita have done in Pleistocene Park, where they're putting large herbivores back on that ecosystem. And in doing so, they're recreating these more robust ecosystems. They get a richer diversity. What happens there is some of the animals will move along the surface and they'll scrape away the snow. And snow is a very efficient insulator, so it keeps the summer warmth in that Siberian permafrost. If they scrape away the snow because they're looking for something to eat, that exposes the surface in some patches to the very cold Siberian winter air. And so you get a much patchier ecosystem. And when the spring thaw comes, you get then a greater diversity of plants that grow back because you have some that prefer this colder, drier sediment and some that prefer the warmer, wetter sediment. And the greater diversity of plants can then support greater diversity of herbivores. And I can see that having these animals back on the landscape benefits the landscape, right? Yes. I've also worked with communities in Tuktoyaktuk in the northern part of Northwest Territories that have already had to move some of their settlements back because of sea level rise. I mean, it's clear that there is habitat change that's happening around the world. And it's also clear that the rate of change is faster than species are able to catch up. Now, I think that what we need are a greater diversity of tools that we can use to help species that are alive today adapt to their ecosystems and avoid becoming extinct. And in some cases, we may need to engineer traits that are now extinct, species that are now extinct, to put them back in these habitats to make those ecosystems more robust and resilient. So how do we get there? The same tools that we would use to bring a mammoth back are the tools that we need to help Hawaiian honey creepers become resistant to avian malaria. The toolbox, the kit of resources that we're developing at Colossal that we need for de-extinction are also the tools for biodiversity preservation. Colossal is a de-extinction company, but we're also a species preservation company. And as we develop these resources, we're making them available to use for conservation, for the purposes of ecosystem restoration for free.
Amazing. So I'm satisfied with that answer. We're not going to do a long form debate on whether we should or shouldn't. I want to dive into the science. So how do we clone a mammoth? Let's start there. You wrote a book on it in, I think, 2015, not a typo, more than 10 years ago, 2015, 16. So you've been thinking about this for a long time. Maybe you can give us a step-by-step. And I'm really interested in where does the technology need to develop in any way to make that possible?
Sure. Well, there's a lot of steps. So I'll just start with the obvious one, which is that it is not possible to clone a mammoth. So that's sort of the punchline from the book.
Yes. So we have to make some concessions, right? We can't have exactly what we want.
Well, it's really because cloning is the process of somatic cell nuclear transfer, the process that most famously brought us Dolly the sheep, but now is used in agriculture quite a lot. So, and you can even clone your pets. There are lots of things that you can do with somatic cell nuclear transfer. But what's critical is that it requires a living cell, right? You can't clone anything. You can't transfer the nucleus of a somatic cell into an egg cell that has had its own nucleus removed if you don't have a living cell.
Right.
And so from the beginning, it's not possible to clone a mammoth because there are no living mammoth cells. So instead of cloning, what we do is engineer the genomes. And this is normally where we start with Jurassic Park. And I think starting with Jurassic Park is fine and it's fair because it gives people an idea of what happens. Okay, so tell me what the scientists did in Jurassic Park. Do you remember?
If my memory is correct, they took the mosquito that was preserved in amber and they found the dinosaur DNA from the blood that was within the mosquito, and then I think they used a chicken, right, to
I don't know that part, but do you remember how they pieced together the dinosaur genome? Because this is where we get stuck with everything.
I don't.
I think that what they did was they took the pieces of dinosaur DNA that they got out of the mosquito. And if you imagine this, like you're a scientist, you're a geneticist, you know how ridiculous this is. They had the physical pieces of DNA and they somehow like stitched them together.
Right.
And where they were missing some, because they were like, well, we can't have all of it, right? They took frog DNA. Do you remember this? They took frog DNA and put the bits in the middle. Well, why they chose frog DNA, I don't know, because
they seem frog-like. Yeah.
Right. We already knew birds, dinosaurs, they're the same. Frogs, totally different, right? So why frogs? I don't know. Doesn't matter. But that doesn't make any sense, right? You wouldn't when I sequence my own genome, I'm not doing it by actually taking two pieces of my DNA and stitching them together somehow, like physically stitching them together.
Yeah.
That's not how we do it, right? So if I extract DNA from a mammoth bone, I will get the whole mammoth genome. All of it is there many times, right? But in tiny little fragments that I don't know how to piece together. So the way that I do it is I take the closest living relative, that's an Asian elephant. Asian elephants and mammoths shared a common ancestor around 5 to 6 million years ago. Fascinatingly, that makes Asian elephants and mammoths more closely related to each other than Asian elephants are to African elephants. They share a common ancestor some 11 million years ago, right? So they're more closely related than African elephants. They already share more than 99% of their DNA. That makes sense to us, just like we do with chimpanzees, et cetera. So we've already got quite a lot of mammoth with a living Asian elephant cell. So that'll give you a hint to where we're going with the cloning, right? Okay, so I have all of these tiny little fragments of mammoth DNA, and using a computer, I can one at a time map them to a reference Asian elephant genome. So we have beautiful telomere to telomere Asian elephant genome that we've helped to produce. It's part of the Vertebrate Genomes Project and will be part of their phase 2 analyses that's going on. Super awesome Asian elephant genome. We can take each of those pieces and obviously using computer, map them to that reference genome. And then you get a reference genome here that has lots of stacks of tiny little fragments randomly selected from the genome because DNA decay happens randomly. And then we can interpret what the sequence of the what the sequence of the mammoth genome is by looking at the consensus of all of those fragments, right? Does that make sense to you?
Yes, we're aligning it to the Asian elephant genome, basically. Yeah.
Cool. Yes. And we can do that for loads of mammoths. And mammoths, unlike dodos, we have loads of really well-preserved samples because they lived in cold places where DNA preserves better.
Yeah.
They are big bones, so the bones themselves preserve better, and the DNA within those bones preserve better. We have mummies, but mummies are not so great for DNA, it turns out. But bones are.
Why, the mummification process breaks it down or something?
Probably because it's microbial. I mean, most of DNA decay happens because of UV light. of freezing and thawing, which the water expands and contracts, and so it physically shears the DNA fragment, and then just microbial decay. So microbes will get in and eat up the DNA to turn it into the next generation, and so that breaks down the DNA. So that mummification process probably sends microbes all throughout the body of the animal, which just chews up the DNA. But anyway, so we have loads of these. We can compare them all to each other, and then we can ask, where are all of the mammoths the same as each other, but different from the Asian elephant? This is similar to what people who are interested in identifying what it is that makes humans uniquely human do in comparison to Neanderthals. Look at all the hundreds of thousands of millions of human genomes that have been sequenced and ask, where are all humans the same as each other in a mutation that has evolved since the split with Neanderthals? What is fixed derived human? Those are the things that are going to be most important to making this human. We do the same thing to identify what it is that is most important to making a mammoth instead of an Asian elephant.
Yes.
Cool, right? So now we have this big list of places in the genome where all mammoths are the same as each other and different from Asian elephants. Still a pretty big list. Right now, that list is about 1 million or 1.5 million different places, and we're still getting more mammoths. And as we get more mammoths, the number will decrease, right?
And when you say places here, how much of the difference is like, so when I did my PhD, I studied these human accelerated regions and ultra conserved elements, very interesting parts of the genome that are very identical except for a couple of changes. But then also you have huge duplications and all sorts of crazy rearrangements that maybe are hard to capture in this kind of setup. I'm sure you can get to it, but I'm just curious on like the texture of those differences and how much is driving what is essentially like, what is the essence of woolly mammoth? Is it, how much of it is some of these big duplications and changes versus smaller changes at a single nucleotide or smaller?
It's a good question. And we can't really answer the question about large duplications because our fragments are all very short, right? But we have a paper with Luva Dahlen's group in Stockholm where we've been able to get long fragments of DNA, some very long fragments of DNA using nanopore technology on some extremely well-preserved mammoths. So we hope in the next 6 months or so to have a lot more information to contribute to structural variation in the mammoth. But until then, we can't see it. So we haven't really been focusing on it, although it's definitely something we're interested in. When we look at, where are these 1.5 million differences, we think about things like how many of them are in genes, how many of them are in introns, especially the first intron, how many of them are enhancers or promoters, are there things that are in accelerated regions that exist in some mammoths or all mammoths? Is it accelerated regions in all elephantids? There's lots of ways to, as you know, to characterize those different things and then think about how we prioritize them to figure out whether they are responsible for these phenotypic differences that we see that we want to bring back when we are talking about de-extinction. And this, I think, is, it's one of the hardest problems in biology right now. And definitely one of the things that the mammoth team at Colossal are interested in. Same is true for all the other teams. Our dodo project, for example, the closest living relative of the dodo is a much smaller bird called the Nicobar pigeon. Really different face shape. So one of the things that we're interested in learning is what genes and promoters, enhancer regions drive changes in craniofacial shape in bird species. And one of the ways we've been attacking that is looking at what genes are being expressed at different developmental stages in birds. We've collected a whole bunch of 3D scans of skulls of birds and are using machine learning algorithms to reduce that 3D image to something that we can then compare to genome sequences, really doing everything in our power with modern technology to try to narrow down on what are the most important changes that we need to make to drive those phenotypes. Because what we want to do is make the fewest number of changes possible to the genome to drive the phenotypic change that we need in order to achieve functional de-extinction.
Yes. And is there a formal definition of functional de-extinction? Like how do you think about that? Because I think we can both agree that it's an impossible task to get to precisely preserved woolly mammoth, at least with the technology we have today. But what we care about, at least in your framing, is we want something that may be able to restore the ecology to what it was before. So how do you think about that end goal and how we know when we got there?
Well, when I say functional de-extinction, I'm really thinking just about that. If I am wanted to bring a dodo back. I want to bring a dodo back because Mauritius and the ecosystems there is missing a large fruit-eating, ground-dwelling herbivorous bird. And if I achieve functional de-extinction, I will replace into that ecosystem a large fruit-eating, ground-dwelling, flightless bird that can restore the ecological interactions that the dodo once had in that ecosystem. And you can see the benefits of doing that from work that's happening in Mauritius right now. They also lost a giant tortoise when the dodo went extinct. And they have replaced the giant tortoise in Mauritian ecosystems, not through de-extinction, but by going to Seychelles and taking the giant tortoise from there and reestablishing that animal on the ecosystem. And they have seen tremendous ecological benefit, cascading ecological benefit from doing this. One of the things, for example, is that they've been doing a lot of invasive species removal, which is really important for us because if we want dodos, we can't have cats and rats and pigs and other things. But they've been trying to remove non-native plant species, for example, doing it by hand, going into these ecological sites and pulling out these plant species. But when they established this giant tortoise population, reestablished the giant tortoise population, what they immediately saw was that these tortoises quickly gobbled up all of the non-native plants that did not evolve with giant tortoises, and so had no anti-tortoise herbivory mechanisms when they were little baby plants. But the native plants that did evolve with giant tortoises had things like serrated edges that the tortoises didn't like to eat, or concentrated toxins that the tortoises didn't like. And so having the tortoises on this landscape has helped them to get rid of these baby non-native plants that are coming back and restore these populations of the native species just by putting that interaction back in place on that ecosystem. They've also seen that the ebony trees, the ebony forest is coming back because the ebony seeds can germinate more readily after they pass through the digestive system of the giant tortoise. So these are all unexpected ecological benefits to restoring these species to the ecosystem. So that's really what I'm thinking about when I think about functional de-extinction.
Yeah, I love that. I have one maybe quick fun question about the woolly mammoth before I move on to something else, which is just really a curiosity of, have you been able to pinpoint the genetic locus or loci that drive the hairiness, the woolliness of the mammoth? Because that's quite a big difference between the woolly mammoth and the Asian elephant, and it has that amazing characteristic coat. And I'm just curious if we know where that's coming from.
It's definitely one of the traits, one of the key traits that we're focused on. And it's both the character of the hair itself, longer, thicker, curlier hair. And I'm sure you saw the woolly mice that we made where we targeted specific genes. You didn't see our woolly mice? Oh, you have to check that out online. They're super cute. Yes. So we identified a couple of different genes that our mammoth genome analysis were suggesting are associated with woolliness in mice. And we didn't put the mammoth, in most cases, in a few cases we did, but in most cases we didn't put the mammoth specific variant into a mouse because, a mouse is not a mammoth. There's a lot of evolutionary distance there, but there were variants in those same genes that led to different woolly features in mice. And so we combined a whole bunch of them and made little woolly mice that are using genes that were identified by our mammoth analysis as being associated with this woolly phenotype, and created our ultra woolly little mice. Did you check them out? You should.
Yeah, I did. I was looking at them in the background. They're very cute.
They also have a mammoth-specific variant associated with subcutaneous fat, which is, of course, another one of the traits that we're interested in is cold adaptation, because we have to take this tropically adapted animal and let them able to live in temperate and even colder environments. So that's another trait we're interested in. But it's not just the trait of the hair itself, it's also the density of hair follicles. So elephants have hair, they just don't have much of it. So we're going to need to figure out what it is. And I'm sure it's not just one gene, obviously, but we do a combination of analyzing the mammoth genome itself and then doing comparative analyses across the tree of life and look at sister lineages that have dense hair versus not particularly hairy, and then do these large scale analyses capitalizing on machine learning algorithms that can help us narrow down on what genes or what regions of the genome we should be focusing on, and then go back to the mammoth analysis and say, is there anything there that looks like it could be under selection in this lineage or fixed in this lineage compared to Asian elephants?
Yeah.
So.
suppose that for any of the examples that we're trying to get to, the dire wolf, the woolly mammoth, anyone else, we can come up with that minimal set of genetic changes between the nearest relative. Is that really the key blocker in most cases? And then you can use modern genome editing tools to edit the nearest relative and get there, or is there some complexity beneath that you're still working on to maybe we can't get quite there?
There's so much complexity, and every species that is a candidate for de-extinction will have a different set of technical, ethical, ecological, societal issues that need to be addressed, resolved, discovered. Scientifically, lots of challenges. Obviously, we're not going to be able to get away with just making 11 changes. We made 20 edits to the dire wolves. That's not going to be enough to transition an Asian elephant into a mammoth. So we have to work on multiplex genome engineering. And so we have teams that are focusing on making lots of edits simultaneously, and then also inserting large and replacing large regions of the genome so we can get multiple genes or even loci. I mean, who knows if we'll ever be able to narrow down to one specific causal mutation, but we know from extended haplotype analyses that this locus is what is driving this phenotype. Let's put the whole thing in then, right? And so driving sort of.
Can you do that today if you find a whole locus that seems to be associated with woolliness, for example? Can you splice that into the Asian elephant pretty successfully?
Well, people have done this with a lot of work with yeast and microbes, of course. But yes, there is technology that will allow us to move big pieces of DNA into and out of a genome. But we, of course, want to move it into a very precise location. We want to do it multiple times. We want to make sure they're homozygous. So there's a lot of work to be done there to make sure that we can drive those technologies. The other thing is, I've talked a lot about cloning. Eventually what we end up with is a living Asian elephant cell that has all of these edits in it that make it a mammoth cell that we then clone using somatic cell nuclear transfer. But that means that we have to then do somatic cell nuclear transfer followed by embryo transfer and gestation in an elephant. And that is advanced artificial reproductive technology that still needs to be developed for that clade of the tree of life. And again, I started off by saying these are technologies we need for biodiversity conservation. We're working with conservation partners in the elephant and rhinoceros world to develop these technologies.
Yeah.
And as we develop them, they will first be applied to the endangered species and make sure that this is working before we use them for mammoths, right? So, but that is an incredible benefit to these endangered species that have us really pushing and providing investment in developing technologies that are useful right now to help endangered species avoid becoming extinct, that are also useful and necessary for de-extinction. Yeah. Birds, now I shifted here. Birds, we can't even clone, right? It is not possible to clone a bird because we don't have access to the egg cell at a stage where we can remove the nucleus of that egg cell and transfer the nucleus of an edited somatic cell into its place. So we need an entirely different approach to modify birds in a way that gets passed down to the next generation along the germline.
Do you have an idea what that is? What is
the
that?
The technology that we're using, that other teams are using. There's a group at the Rodham Institute in Edinburgh that have really been pushing this technology, is to modify primordial germ cells. These are the cells that will eventually become either sperm or eggs, depending on the biological sex of the animal. When the egg is laid of a chicken, for example, you can stick a needle into that developing embryo and suck out some of the blood, and the primordial germ cells are circulating through the bloodstream at that point, trying to find their way to the gonads, which are still developing. You can do that without harming this embryo, and you can stick those into a dish, and you can modify those cells. Right. And then you can re-inject those modified cells into an embryo at that same developmental stage. They will migrate through the bloodstream and establish themselves in the gonads. Ideally, you're doing that in a genetically modified bird that doesn't make its own germ cells. So there's no competition between unedited and edited germ cells for actually making the next generation. And that is our plan for making dodos, to use a chicken that doesn't make their own germ cells, to inject into that chicken edited pigeon cells, right? Because dodos are a type of pigeon that will establish in the gonads. And we will then breed two of those birds together, and that chicken will lay eggs that hatch into a dodo. Doesn't that sound like science fiction? And yet it is my actual job to make that happen, which is, it's beyond cool, right?
It really is fascinating. And you've got this really interesting, almost like a scaffolding of, you got a very long-term research arc, but you have, and it depends on 3, 4, 5, 6 of these things being figured out. But each of those, as you pointed out, has a very near-term application. And so you can ladder this up in a way where you don't have to wait 50 years to see impact, right? You can think about what you can do today and how that ladders you up to the big picture.
Yes. For example, we published it was about a month ago, we put on the bioarchive, and it's now, paper's now undergoing peer review, a recipe to keep pigeon primordial germ cells alive in a dish, which wasn't known until our team started throwing together all these different combinations of all the stuff that you might need in a dish to keep a cell happy and alive. And we finally came up with that recipe, which we can now use for our Nicobar pigeon cells, and have made it public so that anyone who wants to use this work on endangered pigeon species, like the pink pigeon, can use that recipe. So we are developing technologies along the path to our de-extinct species that will have immediate benefit to helping other species avoid becoming extinct. And that is really the thing that drives me.
Yeah. I wanted to ask you about a different concept and aspect of this, and I'm thinking of it because I remember learning about this idea of going after pathogen carrying mosquitoes with gene drives, and there's this really interesting concept of you can essentially genetically engineer at a population level to influence this public health issue, and the common question is, how do we make sure that we have an off switch in some way, or if there's some unexpected impact, that we have a way to control this? And so my more general question here is, how do you think about genetic off switches in these different examples that you're thinking of? Because that's probably another pillar of the scientific strategy, right?
Sure. So you're talking about gene drives in mosquitoes, and I think a lot of the literature about gene drives, and this is passing a trait through a population faster than natural selection would happen. We have these ideas that Kevin Esfeld talks a lot about, like daisy chains, where the drive only persists for a certain number of generations. That's based on the number of links in that daisy chain. So every generation, one of those links breaks, and then when the last one breaks, it disappears. And so the edit will disappear from the population. And so it doesn't just last forever and get out there. I think one of the things that people have asked me is, do you worry at all that your mammoths are going to get out into the population and take over and destroy things? And honestly,
or become an invasive species, basically, right? Something like
so
that.
I don't mean to be facetious about this, but elephants have a 22-month gestation and they've reached sexual maturity at age 14, right? I don't worry about mammoths becoming an invasive species because honestly, it will take an extremely long time for mammoths to build up to even a sustainable population size. And this is terrible, but people already killed them all once. If they really get out of control, it's terrible.
Yeah, I'm not worried about the mammoths.
I'm more worried about these animals actually surviving than I am about them becoming an invasive species. And in any of these, obviously our goal with all of the species except for the dire wolf is to eventually release them into the wild so they can become a free living population. Yeah. But they will always be managed in some way, and it will be very staged. Like those initial populations will be very heavily managed. We want to make sure that they're healthy, that they're interacting with the ecosystem in a way that is predictable, that we've actually thought about all the potential risks of releasing them into habitats. And obviously, anything that we do is under regulatory purview of different environmental agencies, no matter where they are. So there will be lots of people watching to make sure that things are going according to plan. And there will have to be plans in place to make sure that it can be stopped if something does happen that causes it to get out of hand.
Yeah. And I guess in my like, the way I'm thinking about it very simply is there's probably a big spectrum between, if you think about mosquitoes on one end, where we can't track them down. You'd have to come up with some kind of
system. And they make hundreds of millions of eggs, right?
So, yes. And then you got giant mammals on the other end. And as you work through each of these cases, there's probably things in the middle where you say, okay, if the dodo gets out of control, here's the plan, right? And you have a risk-based approach to it that you figure out. Yeah.
Absolutely. It's a really important part of it. And for each of our species, we actually have local advisory committees. So we have, for example, for the thylacine, we have a Tasmanian advisory committee that's made up of local people, business leaders, politicians, environmentalists, et cetera, that are all working together well before we have anything to release, to develop all of these plans and to really ask the hard questions about how many do we want, where would we release them, how would we watch them, what is this risk mitigation plan that we should have in place, long before we actually have the animals that we're ready to release. And I think it's really important to have these conversations and to have them out loud, right? Which is one thing I really appreciate about Colossal. I mean, everybody's heard of Colossal. Everybody knows what we're doing. Some people are really mad about it. Some people are really excited about it. But there's nobody who can say that we're doing anything in secret.
Yes. I think that shouldn't be overstated. It is really important, right? And I think that you have lots of different frameworks to make these decisions. My sense from the outside looking in is you do a lot of work with the communities that you're planning these projects with, right? And someone has to pay for this as well, right? So it's probably driven by collaborations with large scale conservation organizations, nation
Absolutely.
states.
And all of that information is on our website, all the conservation organizations. I mean, a good example of this is the most recent project we launched, which is the South Island giant moa, which is a project that is led entirely by the Ngaitahu Research Center, which is one of the largest Maori groups in the South Island of New Zealand. So really working with our Indigenous partners to make sure that this is what they want, and they are driving decision-making about this project. And I think that is the way this has to be done.
Yes. If you think about the goals you have, I don't know what time frame you're thinking on, but I would love to hear about the 2 or 3 key steps that are missing right now, and where this could be at the understanding the fundamental genetic differences. It could be at maybe the more translational stage where you're going from that to bringing things back to life, or it could be something else. I'm just really interested in, if you're looking out 5-10 years, what are the big steps that if we can crack them, we'll live in a really different place?
I think, that's a question I've never been asked, and I really like that. So there are so many things. If I pick each of the different components of de-extinction, they each have fundamental technological discoveries that need to be made. So multiplex genome engineering obviously is something that we really need, like big multiplex. And as we get to that has cascading benefits, even for human health and how we think about genetic modification to fix genetic diseases, somatic level, whatever. We need advanced artificial reproductive technologies for species that aren't agricultural species or mice, lab mice, right? And all of that for across the tree of life needs to be developed. Birds, there are so few resources, approaches, tools for using biotechnologies for birds. So that is a huge development. And, obviously mapping genotypes to phenotypes. I mean, we have so many genomes that have been published right now, but these are still like phone books missing the names, as people like to say, as an analogy. And so how do we figure out? And I think this is a place where AI is really going to have a tremendous impact over the next 10 years. How do we figure out what genes, loci, promoters, structural changes lead to the differences that we see in morphological, behavioral diversity across the tree of life? Behavior is obviously a really hard one too, because it's obviously multigenic and environmentally influenced. So how do we get to these things? There's a lot of work in ecosystem reconstruction and stability, and understanding how perturbations to ecosystems that happen naturally or that happen because of human intervention, accidental or on purpose, are impacting the long-term trajectory of ecosystems. That's a really interesting and hard thing. And then obviously with genome engineering and gene editing of any type, global regulatory mismatch, right? My dogs are mad about that too. Yeah. How do we bring together the governments across the world that disagree about what we can do, what we should do, how far we can go about these things, and create a framework that enables research, but in a safe way? I mean, obviously we have to acknowledge risks associated with what we're doing, but I think one of the things that we fail to do as humanity is acknowledge the risk of not allowing ourselves to explore what these technologies can do for us. We know that we are living in a period of a biodiversity crisis, and we know that a lot of that is because of changes that people have made to habitats around the world. if we say, oh, these are too scary, we're not going to do that, we don't like the idea of GMOs, and we could talk for another 2 hours about how messed up GMO regulation and converse. I mean, we have non-GMO salt, right? It's not it's a rock. It's got no G's to M. Like there's so much that's messed up in that world, right? That we really are going to have to. We're going to have a, I don't know, come to the table moment across the globe, and
the secular version.
How to create a path forward there. But like I say, I will do my part by continuing to have these conversations out loud, by openly disagreeing with people and being willing to have a civil conversation in that space, and talk about why it's important that we're doing the work that we're doing at Colossal, what we're bringing to the table, how it's not in competition with traditional approaches to conservation, but relies on those and is a yes and moment rather than an either or. Because I think that if we don't do this, we're just going to see an acceleration of the extinction crisis that we're in. And I want to be part of the solution.
Thank you. I mean, this was an amazing conversation. I learned a lot. It was a lot of fun. You're an amazing storyteller. I think, like we discussed at the beginning, you've weaved together the hard science and the storytelling in an amazing way. So appreciate you sharing that with us.
Thank you.
And everybody, thank you as always for listening. We will see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate it if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.